So this next section is about data formatting and preprocessing. The first part is about data formats that you might see a lot in this particular field. So there's a lot of outputs and data formats that are currently being used in the spatial omics field for both storage and dissemination of this information. The following are some that we commonly work with. For Giotto, a lot of the data wrangling task is just to get the information out of these formats into native R formats and then wrapped as the Giotto sub-objects, which are compatible with the Giotto object. So general formats are like things like .csv or .tsv. So very standard delimited file types. These are values that are separated by commas, tabs. You can read in these with a wide array of different possible functions that you could use. So there's like some from utils, reader, package, and also data table. They're very easy to use, but the larger files are also hard to scan through, and they don't really, they may have like interesting ways to parse through their information or figure out what information they contain, but they're very widely compatible. For matrix formats, we regularly see that 10x provides their cell feature count matrices in both the MTX, which is also known as like matrix market or MM format, and .h5 formats. So the MM formats, the matrix market formats come in a zipped folder where the inside is usually in this structure. You have a barcodes.tsv.gz file, features.tsv.gz, and then a matrix.mtx.gz. So this format by itself, which is really just this core file over here, does not carry dim names for the call names and row names. So they're stored in the TSVs over here, or barcodes, which are the cells or observations, and then the features, which are like variables, genes, or protein expression. So this is an example of the barcodes.tsv from a Xenium dataset, for example, where all of it is just barcodes for each of the cells. You can see that there's like 162,000 cells in this particular snippet of it. And these also don't come with an actual column name for each of these. So you would read it with a header equals false. The features.tsv from the same Xenium dataset, you have a couple of different columns over here. So you have like the ENSG ensemble ID over here. And then you also have the actual gene names that we like to use more often. And the third column over here is actually for like which particular type of gene or which particular type of feature it is. So they can distribute more than one expression type inside of these. And what we like to do is we have an actual like reader function called get10x matrix and get10x matrix h5 that accesses both of these two formats, parses through these. And if you want it to, it can also split up this information into more than one matrix based on third column over here. So yeah, that was the third file in there is the matrix.mtx. That contains the actual sparse matrix values in triplet format. And the H5 format is very similar, except instead of having like a zipped folder structure like that, it just is a hierarchical format that contains all three of those items in the same file. Moving on to tabular formats. So recently, .parquet has been used a lot more. It's a great format for storing a lot of large amounts of table information and providing fast access to only portions of the data at a time. 10x is using this format for things such as the transcript detections from Xenium or the polygons information. I think VizGen is also doing the same with their polygons data now. They can be opened and work with using arrow and dplyr verbs. And currently, Giotto extracts information from these files and then converts them into in-memory data tables or Terra spat vectors, depending on what data they actually contain. For spatial formats, there's .shp and .geojson, which are commonly seen ones. These are common formats for polygon and point data, and they're commonly used as exports from segmentation software such as QPath. Create Giotto polygon, which is exported from Giotto class, and the more specific create Giotto polygons from GeoJSON for GeoJSON specifically can also be used for reading these in. So I think library in Giotto, already library Giotto class, but I'm going to run that anyways. And these are just some toy examples from that are stored alongside Giotto class. We actually use them for unit tests, but yeah, that's just me pulling out a file path from Giotto class. I would create the Giotto polygon from that SHP that's over here. And you can see that this is what it looks like when you take a look at the print or the show function. It is labeled as test now because of this name. And also I can just plot it and this is what comes out of it. So that's what was stored inside of the SHP file. For mask files, TIFF files are used. We often see like TIFF files that are used as mask files, where the actual integer values of that image will encode where a particular annotation is. So we have create Giotto polygons for mask, which guesses whether the image is, either single value or multi-value mask. Cosmics is one example of a platform. Vanishing Cosmics is one example of a platform that distributes the polygon information through a series of mask files. This is just another toy example. So this is a mask with multiple values, just to show a quick plot of that. This is it loaded in as an image at the moment. So I'm using Terra to plot this particular file. I am coloring it with HCl color 7, which basically defaults to Viridis. I'm pulling 7 colors out of the Viridis color palette, and those colors are then mapped like integer values. So each one of these with a different color, that's a different integer value from the mask. The white is just no value. So if I now put this into create Giotto polygon, this will create a Giotto polygon from that image. And that doesn't work so well with a markdown. This is the show function from Giotto polygon. You see the spatial unit, you see like the internal contained information is a spat vector, and you see that the polygon IDs are over here. And then furthermore, I can just plot it, and the colors are the same as shown previously, meaning that we actually do read each of the numerical values that are encoded, and it does get added. We add our polygons in the correct order based on the encoded values. This is just another example for situations where you might have like single values. So it's like either a 0 or 1 value inside of your mask, and then any areas that are not connected to each other get different annotations. So in such cases, we would run it, and well, it auto detects it actually. So I would run that again, this time with M2, which is this image over here. And then I can plot it, and it shows that now these are split up based on like which annotations which set of pixels were actually touching each other. For images, most images are openable using create Giotto large image, which wraps Terra Rast. This allows compatibility with most common image types. Recent and non-geospatial related image formats, though, are not as well supported. So one example is OmiTIFF, which 10x is using now for large image exports from Xenium. And for these, we have a converter that we export from Giotto class called OmiTIFF to TIFF, which converts them just into base TIFF files using the Python TIFF file package. OmiTIFF metadata can also be used to extract and access the associated OmiXML image metadata. I'll show more about this when it comes time for the actual Xenium tutorial. For JSON-like formats, these are formats that are ones that can be read in with JSON-Lite package, read JSON, and then coerced into more like list-like or tabular structures. 10x uses these .jsons to support scale factors information in Visium datasets, for example, the Xenium file format. So you usually see them as like experiment.xenium. You can also open those as JSON-likes. And hierarchical formats. So there's a lot of data in spatial omics analysis, many types of data. Hierarchical formats basically provide a way that you can organize multi-type and also complex data and store and distribute them. So in R, you can open these with either HDF5R or RHDF5. One's on CRAN and the other's on Bioconductor. They do operate in different ways. I think this one's like R6 and this one's an S4 format. The complex nature of these formats and also the fact that they are just a storage format and not really an organizational specification means that what data and how it's stored and represented exactly can be pretty different. .gef and .bgef, which StereoSeq platform exports, are also .hdf5-like formats and can be opened with these packages as well. .h5ad is a specific flavor of these file formats where they actually follow the AND-data framework. So there's a lot more of a common structure in how these data sets are stored. Particularly well-known would be like AND-data with ScanPy. Those are ones that we actually do have a conversion function for. And AND-data to Giotto and also Giotto to AND-data are the interoperability functions that we use for that particular type of H5AD. .zar is another hierarchical storage structure. However, currently the R native support is still being developed. So next is pre-processing. The most common types of raw data that are needed for Giotto objects are expression matrices, centroids information, spatial feature points, polygons, and expression information. I said that twice. Evaluation of input data and conversion to compatible formats will then happen inside of Giotto's like create functions that Giotto exports. So these are create functions for create functions for sub-objects. And there's one of these for each of the sub-object classes. I'm just going to show some examples in a second. For expression matrices, not much processing is needed for these. All that you really need is a data type that you can coerce to matrix or the matrix package classes. And dim names should also be added, preferably. The columns should be cells or observations, and rows should be features or variables. So here I'm just making a example matrix. It's 10 actual values spread out across like 90, or spread out across a 10 by 10 matrix. The rest is zeros. I want to set a couple of row names and call names. So now that it looks like this, and then I would just run create expression object, and out of that I would get a Giotto sub-object that looks like this. So this is an expression object with a name of test. Of course, you can set those in here by setting name, new matrix instead, and I can do that. The default spatial unit and feature types are going to be cell and RNA. Spat unit basically describes what length scale you're working on, and feature type is just what type of feature information you have. So here is RNA information, or that's the default anyways. And it's just a pretty example of, or just a pretty print of the matrix object that's inside of it. And here are just spatial locations. So I'm going to set a seed just so it's reproducible. And after that, I'm going to make a quick example data frame of XY values. The call names don't really matter too much right now, although it will actually look for things called cell ID, STIMX and STIMY, I believe. Those are like particular column names that it's going to look for. If they're provided, it's going to automatically take those values as the expected columns. But once you have this as XY, you can create SpatLogs object, create a spatial locations object. It looks for the first non-numeric, first non-numeric column and sets that as the cell ID. So that was A, 1 to 100 as character. So those are set as the cell IDs, as you can see over here. And then after that, I did a bunch of random values for B and C. So those are incorporated now as first X and then Y, which we label as STIMX and STIMY. And you can also plot a lot of these sub-objects, particularly the spatial ones. So this is just a plot of it that you can directly see. And then next is, for example, of 3D data. So I wrote this, but I forgot that most people were, we didn't actually include RGL as one of the expected packages. So by default, it is showing you with RGL, which not everybody has installed. But this is what happens when you create it with 3 columns of numerics instead of just 2. And yeah, so that was creation there. Just forgot to show the show function. And next is Giotto points. So Giotto points are very similar to the spatial locations. These sub-objects instead wrap a teraspat vector object. So these previous ones over here, they wrap the data table SL XYZ. This is the actual data table that's contained within. What I used is just like an empty bracket. I'll mention more about that later. But yeah, that's the internal structure over there. Back to Giotto points. So these wrap a teraspat vector object, and if tabular information provided, what you need are X, Y, and feature IDs. So additional columns are also kept as metadata information. Just going to make an example table. So this now has a set of IDs, which are character, X and Y values, and then some random metadata that you might have. I would run create Giotto points on that particular table, and it creates a set of G points for me. I can plot this using raster false, which basically tries to show it without rasterizing. This is the actual data itself in like vector format. And then we also have, by default, it's raster equals true, which takes a similar approach to scatter more, where you actually rasterize or bin the points so that it produces the plot faster. Of course, you're not going to see too much difference right now because this isn't that many points, but that is what we implement in the backend for that. And I also set dense true on top of raster true, which is by default in there. So that allows it to actually plot it based on how many counts were in each of these bins when it rasterized the points information. So it's density information. It's a density plot now. And how the show function actually looks is feature type, RNA, contains spat vector data, a set of dimensions, this meaning however many actual records or different transcripts are being kept in here, and how many metadata columns there are. And then this is the spatial extent, which is like the spatial enveloping box for it, and just a preview of what metadata information it contains. Most importantly is like feature ID, and also feature ID unique, which is something that we add ourselves actually, so that we can keep track of a unique ID for each of these features in the Giotto points. Next is Giotto polygon. So polygon information is often provided as a known spatial format or as image masks. You can read these in as shown earlier with the GeoJSON or like the mask images, but they can also be provided as numerical values. So this is actually the case for Vision Merscope and also 10x Xenium outputs. And both of these now use Parquet to provide the cell barcodes and XY vertices associated with them. Actually, yeah. Hex vertices, this is just a function that we export from Giotto class. It's a convenient function to generate a set of points in a particular shape. That's just what that is. And then just a set of spatial locations. So I'm just setting up a set of vertices and also cell IDs to better explain how this function works. So now we have basically a set of points that define a particular hexagonal polygon with a radius of 1 and a set of spatial locations. And we use polystamp, which is also from Giotto class, which basically takes this set of vertices and stamps them across each of these XY coordinates, and also transfers over these cell IDs. So random hex, that generated 10 polygons because there are 10 spatial locations we just stamped across. And then we can, I just show that first. This is the raw data that would go into create Giotto polygon when it's a tabular format. You have X vertices, Y vertices, and then a set of polygon IDs. So you'll see that these are repeating for multiple values in X and Y, and that just means that these are all associated with the same polygon or intended to be. We create it and then can directly plot it. So yeah, hexagons stamped across random locations. Next is just some sub-object utility functions that we can use. So as mentioned earlier, the Giotto object is hierarchically organized, first by slots that define their sub-object information type. So I have like a previously made example object. I think this was the VizGen one. But just to show you, there's like a lot of different slots underneath it. Most relevant are things like images. You can put images, you can put the nearest networks information for networks you define across dimension reduction data, spatial enrichments, spatial grids, spatial networks, feature and cell metadata, spatial locations, and expression information. So it's pretty clear where everything is actually stored inside of the object, and then you can further dig into it by using $signs, because these are like lists underneath that contain where that information is. So first, usually by spatial unit and by feature type, and then furthermore by like object name, if there's more than one object that is going to exist at that nesting depth. So this is just the spatial unit cell, feature type, RNA, and then the raw expression, and then you get the sub-object. Of course, there's also actual dedicated accessor functions, but I just wanted to show like how explorable the Giotto object is. A lot of the sub-objects are also tagged with metadata information. So like feature type, spatial unit, those allow them to find their place inside of this nesting very easily. And there's also some common functions that the Giotto objects, sub-objects will respond to. So you have spat IDs and feature IDs. These two particular functions are often used in order to find the spatial or feature IDs of an object. We previously made SLXY, which is just this SpatLux object. If you look for the spat IDs, that returns this. So these are the cell IDs that were associated with this. And then if you were to do the same with the Giotto polygons, we set them earlier as like just A, B, C, D, E, F, G. And then for feature IDs from Giotto points, you have this. That's just to show you how you can like easily programmatically access this kind of information from the sub-objects. And then bracket subsetting and extraction. So a lot of these sub-objects also respond to indexing with brackets. But since many of them are wrappers around underlying data structures, empty bracket calls will also drop the objects that contain data structure. So that's something I showed earlier with the Giotto or with the spatial locations object, but here it is with the Giotto polygon, where you can first index, or sorry, I'm actually going to go through like indexing first by row. So this is looking for like the first and second records only, whereas the whole thing is 7 different records, 7 different polygons. And then you can also index by ID instead. It's just different ways you can work with this particular Giotto polygons object. And then after that, this is dropping it from the Giotto sub-object to the internal contained information. So this is from G points. Empty brackets will give you a spat vector from G points. So this is the Terra object that it contains. Spat unit, feed type, object name, and prov are also all generics that work on the metadata of the objects. And these work both to access and also replace that information. So feed type of X, RNA. You can change the naming of the object name, spat unit name. And now those are updated to raw2 in aggregate. And because the sub-objects are already formatted for Giotto and can be directly added to the Giotto object, we can use the set Giotto generic in order to directly add them. So this is an empty Giotto object. It contains nothing at the moment. I can directly set it using that. And X again was this particular sparse matrix or this particular expression object that we just edited. And it tells you that it's just set it to aggregate and it has the name of raw too, just as we specified. And that's it for this particular section as well.